Hi, good morning everyone. Today we'll be talking about this is for the class of BSc Biotech semester 4 biochemistry. We'll be talking today, girls, with regulation of amino acid biosynthesis. The first irreversible regulation of amino acid biosynthesis. First thing is we have to biosynthesize. We have already talked how amino acids are biosynthesized. There were different pathways which were involved. We had dealt with all those different types of enzymes which are involved with that. So today what we will be doing is we will be talking about the regulation of amino acid biosynthesis. Now the first irreversible reaction in biosynthetic pathway is called as the committed step. The first reaction which is there, say A is converted to B, that is called as the committed step. When A will get converted to the B, that is called as committed step, and usually in a very important regulatory site. So the conversion of A to B is the most important regulatory site which is there. So you have to find out the ways with which you can go and control those things. A linear pathway, then you have got a final there are different examples with which you can go in for the controlling of these things. Feedback inhibition is an example here. Say A gets converted to B, B gets converted to C, C gets to converted to D, and D gets converted to E. Now this E which is produced in the last is the one which goes and control the conversion of A to B. So it inhibits the production of B from A when there is a large amount of E which is present. Example is threonine to isoleucine. Example I've already told. So in this case what happens is threonine being reduced to isoleucine at the step A to B conversion. Isoleucine acts as an inhibitor itself when the amount rises, and there is a stoppage of production of isoleucine from threonine after a particular time. Then you can either have two pathways which can take place. In case of a branched pathway, the regulation takes place by different four methods. First one was a straightaway method. When you have got a linear pathway, when there is a branched pathway, you can have different types of regulations, around four types of regulations which take place. First, in case of branched regulation, is called as number one is called as the sequential feedback control. In this mechanism, the first common step, say conversion from A to B, is not inhibited directly by the end product. Say you have got a branched pathway in which A gets converted to B, B gets converted to C. Now this C gets converted to either D or it gets converted to F, D gets converted to E, and then it finally gets converted to Y. Whereas the F gets converted to G and G gets converted to Z here. Now you've got two end products. One is Y, the other one is the Z here. So what happens in this case is I have got a mechanism which is there. Either Y comes and stops the production of C to D, or Z comes and stops the production of C to Z. And C, when there is an accumulation of C, this C goes and stops the production of A to the B, whereas that portion which is stopped is by the sequential feedback. That means one product stops the production of C, say Y stops the production of C, and C stops the production of B. So there is a sequence which is happening here. Example is PEP and erythrose 3-phosphate. This is an example which is there. Shikimate is one product which is formed when PEP and erythrose 6-phosphate they combine up. Now this shikimate gets converted to crosmic acid. This crosmic acid further gets converted to anthrylate and tryptophan. Tryptophan is a very important amino acid. Whereas crosmic acid can also get converted to prephanate, and prephanate gets converted into tyrosine and phenylalanine. Now tryptophan, when it gets accumulated, what happens is in this case it goes and stops the production of crosmic acid. Crosmic acid, that is stopped when there is a larger amount of crosmic acid which is there. That is what happens is it goes and stops the production of shikimate by inhibiting the production of PEP and erythrose 3-phosphate. So that particular critical point, that is where the stoppage takes place. This mechanism operates in the synthesis of, I have already talked about, especially in case of aromatic acids in Bacillus subtilis. It's a microorganism. Also blocking of one of the products affect the pathway of the other. If one product is blocked, the other pathway is stopped here. Then you have got a differential multiple enzyme control. First one was sequential, the second one is the differential multiple enzyme control. Here the first committed step is catalyzed by either of the two enzymes. You can have two enzymes, the first committed step, either of the two enzymes. Say A is converted to B either by enzyme say L, or it is converted to B by enzyme M. So there are two types of enzymes which are there. Here what happens is, now the one part that is say L part, it is controlled by, when you've got a branch chain happening, by Y, whereas the lower part, say, that is of the M, that is controlled by the Z. So the committed step is catalyzed by two. I'll repeat one more time. The committed step is catalyzed by two enzymes, not one here. So the one final product inhibits one part of the enzyme, the other enzyme is generally stopped by the other end product which is there. The condensation example, we can always come here across an example. Example that of condensation of PEP and erythrocyte phosphate is catalyzed by three enzymes, not two even. Sometimes by three enzymes. One is inhibited by phenylalanine, the other one is inhibited by tyrosine, and third by the tryptophan. So that is a sequence that is here called as a differential multiple arrangement. Then you have got the concerted feedback control. Again, a very important part. The first committed step is inhibited in high quantities of both Y and Z. The first committed step. High quantities of both Y and Z. When the amount of Y and Z is too much, what happens is simultaneously they are present. They stop the production of A to B. So that is something which is there. So Y and Z's amount is very critical here. When they reach a particular point, the inhibition of these two goes to the stoppage of production of B from A directly to the B. Example here is that of bacterial aspartyl kinase by tyrosine and lysine. Aspartyl kinase, which converts, it's a basic enzyme which is present, that is being stopped by tyrosine and lysine. So in case of the biosynthesis of tyrosine and lysine, aspartyl kinase is stopped when the production of tyrosine as well as lysine increases beyond a particular point. Then is the cumulative feedback control. Now this is sum total of two. The first committed step is partially inhibited by each of the final product. Partially inhibited, not 100% inhibited. Some parts are 70% by one part, one particular product, 30% by another particular product. A conversion to B is inhibited by this way. This feedback control is present in case of glutamate from glutamate, glutamine from glutamate, ammonia as well as ATP is being utilized here. So that is something which is very important here, that 70% one part, say Y is 70% and Z's 30% is the one which controls the production of C to D, and ultimately the amount of C increases and then you have got the production stopping from A to B. So this is basically a cumulative feedback control, that is sum total of two parts gets cumulated up, and what they do is they go and stop the production of your concerned microorganisms, the reaction which is there, that is A to B. So that is what we'll be talking today. Thank you very much. I'll be sending you the notes as well. Thank you.